Hello, everyone. Welcome to today's live broadcast entitled Predicting Viral Clearance Through the Use of Non-Infectious Minute Virus of Mice and Retroviral-like Particle Surrogates. My name is Jeannie Linking Northrop, and I'm Managing Editor of BioPharm International. I'll be your moderator for today's event. We are pleased to bring you this webcast, sponsored today by Cygnus Technologies. Cygnus Technologies is the biopharmaceutical industry's partner in host cell protein and other process-related impurity detection, as well as analytics, and in the innovative viral clearance solutions. Cygnus helps companies that develop therapeutic proteins, vaccines, antibodies, plasma derivatives, and gene therapies ensure the safety of biotherapeutics prior to those human trials, regulatory approval, and commercial release. Before we begin with today's presentation, I have just a few important announcements I'd like to make. First and foremost, this webcast is designed to be interactive. We have a great presenter on today, and we encourage you to ask him questions throughout the event. You can submit your questions by typing them in the Q&A box, and that can be found at the bottom of your video player. You can enlarge the slide window by clicking on the small icon in the bottom right corner of your media player. Please note that all slides will advance automatically during the event. If you experience any technical problems viewing or hearing this presentation, please click on the question mark help widget, and that's located in the top right of your presentation window. Now I'd like to introduce today's guest presenter, David Setlin. David is currently the Senior Director of MockV products at Cygnus Technologies. He first founded MockV Solution in 2013 and was CEO until its acquisition by Cygnus in 2020. David's lab is dedicated to establishing a series of BSL-1 compatible viral clearance prediction kits. These kits aim to support scientists and benefit downstream purification processes during the development, characterization, and validation stages. David, thank you so much for your time this morning. Please go ahead and feel free to get us started with your presentation.
Thank you, I appreciate it. Just one housekeeping note of my own. That picture of me there is a little outdated. I no longer have a mustache, that's all. So thank you all for taking time out of your day to attend this webinar. As Jeannie mentioned, I'm David Setlin. I was a downstream purification scientist. I worked at a company called Human Genome Sciences, working on all the different process steps to purify monoclonal antibodies from protein A all the way through the polishing steps till tangential flow filtration at the end. And in that capacity, I performed quite a number of viral clearance studies, always at a CRO. And while doing that type of work, I was often disappointed by the lack of ability to perform predictive testing or accumulating any relevant data leading up to that expensive and all-important CRO-led study. So as Jeannie mentioned, I started a company called MockV Solutions in 2013 with the predicated on the possibility of commercializing a kit that would enable downstream scientists like my former self to gain insights and information to help them along the way leading up to that CRO-led viral clearance study. So, and as she mentioned, the company MockV was acquired in 2020 by Cygnus, and now I'm a senior director there, just continuing the work that was begun with MockV, now with a little team under myself. So if you're in the downstream or just in the biopharmaceutical space, you're probably aware of Cygnus Technologies. Cygnus pioneered the impurity kits that we all use in the downstream space to develop and optimize our process steps. Our founder, Ken Hoffman, began this back in the 90s. They launched the first generation host cell protein kits. And along the way, you can see Cygnus has been very innovative at addressing the other types of impurities that we as an industry rely on, such as host cell protein for the different cell lines, host cell DNA, endonuclease was something that was launched more recently. If you're in the gene therapy space, you're probably aware of that product. And orthogonal services too, to really help round out the impurity space, such as extraction affinity and mass spec services as well. So if you're not familiar with some of these orthogonal services that are offered, please check out our website. Of course, my talk will be focused on the viral clearance product line, which Cygnus acquired from MockV in 2020. And so the way that this talk is outlined, first, let's discuss the background, the issue that, at least that I was encountering as a purification scientist, and then the solution that was presented through these MockV, Cygnus MockV kits. So that'll be a little more conceptual, the background of the problem and the solution. And then we'll get more to the nitty-gritty of how we are actually solving this through the use of two different types of particles. One is a retrovirus-like particle, and in that section of the talk, we'll review the characteristics of these particles, how we make them, why we believe in them, why we think they're good markers to track clearance. And then we also need to discuss how we analyze them. The particle is only as good as the analytical method. So we'll discuss that in some detail. And then we'll finally review some RVLP data that was accumulated prior to the launch of the RVLP kits. And we'll discuss some services that we offer related to the RVLP space. We'll shift gears at that point and discuss the parvoviral offerings that we have in the form of this MVM MVP kit. It'll parallel the discussion for RVLP. We'll talk first about the particles, how they're made, why we think they're good surrogates, and the evidence to support that. And then we'll shift to the immuno qPCR assay that we use to quantify the particles. And again, some data sets that we've accumulated over the years. We'll touch base at the end with some additional services offered to cover viral clearance. So as Jeannie mentioned, please do put any questions into the box. This is a novel concept, and it's different from the first 30 years of the biotech industry where we're going here. So any concerns or questions, just drop them in and we'll address them at the end. So, okay, returning to the background. As many of you know who work in the downstream space, biopharmaceutical process development, especially downstream process development, requires a lot of resources in terms of capital, human resources, personnel required, the years it takes to develop and optimize a downstream process. It's a massive effort. And along the way, we as an industry rely heavily on impurity removal kits such as those that Cygnus offers. Host cell protein kits, host cell DNA kits, protein A or other leachets that come off of the resins or other techniques that you use to purify your product. Imagine doing the work of selecting the proper separation techniques and then optimizing those techniques without these available, easy to use, cheap and effective analytical tools. It's really what helped get the industry going to the extent that it has and made it the success that it is. So what was missing from this toolbox, though, was any kit that enabled the detection of virus. And the reasons that why this was missing from the toolkit were obvious enough, but we'll review those in a couple slides. First, however, let's talk about viruses themselves and why we're even concerned with them. The biopharmaceutical industry has a stellar track record of avoiding contaminations at manufacturing scale, but there have been notable contaminations at manufacturing facilities going back to the 1980s. Genentech had one or two. I think a parvovirus was the culprit. And then most infamously, perhaps, was a company called Genzyme up in Boston, whose contamination led to a shutdown of their manufacturing plant for about 6 months. And that was a commercial product that was being produced at that plant. So you can imagine the impact of patients, and not just patients, but the company as well. Genzyme never really fully recovered from that event, and Sanofi was able to acquire them. And it was pretty much a catastrophic event for them. So viral contaminations do occur. The two different modes or methods, modes of transmission into the facility could be through raw materials, which includes cell lines. We'll discuss that in a little bit, but also through exogenous events, through the human interactions. We do the best we can to make these GMP clean room, gowning procedural safe facilities. But at the end of the day, you have humans and you have product, And more often than not, these are exogenous events, especially the ones that occurred to Genentech in the early days of the industry, where it was supposed that mice were, fecal matter of mice was the route that this parvoviral contamination occurred through. So kind of creepy to think about, but that was the smoking gun. So because of the possibility, we're using mammalian cell lines to produce, 70% of the world's monoclonal antibodies right now. So viral contamination is certainly still a possibility. And because of that, regulatory agencies around the world require proof that viral clearance can be demonstrated for the downstream process steps. This occurs before a phase 1 study in the United States, that's an IND enabling study, and before commercial approval of a drug. And in the States, again, that would be a BLA filing. So at 2 points, in the IND filing package and in the BLA filing package, those are 2 times where the data is required to demonstrate viral clearance. The regulatory document that governs this is called the Q5A guidelines, and we'll discuss this in a few slides coming up. There was a draft revision of the guidelines in late 2022. It was the first revision since 1999, and we'll talk about that in a few slides. The only way to conduct a viral clearance study historically has been to contract a CRO that specializes in the propagation and spiking of these viruses into the downstream process on a small scale. So if you're new to the space or a little unfamiliar with it, basically you scale down your process, you typically bring your process and your people into the suites that these CROs have, which are BSL-2 or BSL-3 rated, and the CRO personnel help you conduct that spiking study in that suite. And for a typical CHO panel for a BLA, it's standard to run a retrovirus in the form of XMELV, a parvovirus in the form of MVM, or sometimes a canine virus, but more often than not, it's a mouse minute virus. And then to round out the panel, you have a Rio3 and a pseudorabies virus as well. Let me go back 1 second. I highlighted this panel as is pertinent to CHO, but the risk assessment for what viruses are relevant for your process is really dictated first and foremost by your cell line. If you're using a hex cell line to make a gene therapy product, that's a different, it may require a different sort of risk assessment, which could lead you to a different panel. And we can discuss that in the Q&A if anyone's interested in exploring that a little further. So the way, I hinted at this during the last slide, but the way a viral clearance study is performed at a CRO is propagating live virus. And because of the complexities involved with that and the specialized skill set that the personnel need to propagate that, this work has to be done at the CRO in the BSL-2 or BSL-3 suite. And the world's capacity to host these types of spiking studies is really a limiting factor. It used to be a 6-month, time window. If you wanted to conduct a BLA enabling viral clearance study, you may have to organize this with your CRO 6 months in advance of when you want to execute the study. But what I'm hearing nowadays is that that's really been extended out to 9 months, even beyond. So that's a real issue. In addition, there's the expense of conducting these studies. For a BLA enabling study with that 4-panel CHO line, you could be pushing $400,000, $500,000 to conduct that type of study. So the expense, the logistics, the waiting, and also the turnaround. After you conduct your study, it's usually a plaque-based assay, and then the report writing that follows. And you're looking at waiting about a month before you can look at your results and see how well your process steps do. So there's all of these complexities and hurdles. It is the gold standard for your BLA, your IND. My talk is not trying to discourage the use of conducting these studies. They are necessary, but when you're in the mode of process development and process characterization, it's not a palatable option to wait 9 months, spend $100,000 and wait a month after that for the report. That's where we needed something better, and that sets up where the solution comes into place. The last thing to note is after you've done years of process development and characterization, it's really an inopportune time to discover if your process steps are not performing up to the standards that you wish them to be. For example, if you are using an anion exchange column in your downstream monoclonal antibody process, you're expecting effective, robust clearance, maybe to the tune of 5 to 6 logs of removal for that one process step. And you've done all this process development characterization work leading up to this validation study. If the validation study is the first time that you're challenging your step with live virus, it's a bad time to learn that that step's not effectively giving you 4, 5, 6 logs, which happens. It happens from time to time, and it's a nightmare. You typically have to go back and redevelop that step just to the point of boosting that clearance, And you may have to take a yield hit. You may have to, that extra log or two of viral clearance may be at the expense of your yield. And unfortunately, that's what you're going to be validating, that set of parameters, which locks you into place going into your manufacturing conditions. So in the 7 years that I was at Human Genome, this happened to our group maybe 2 or 3 times. And it was enough that led us, it led me at least to look for a better way forward. And so now let's talk about that. So of course, viral clearance studies require virus. And if you were to come up with a kit to skirt around the necessity of the CRO, you would have to have a surrogate, a non-infectious surrogate, replace the need of that virus. And that's what we do here. So we are replacing the live virus with a surrogate, a BSL-1 compatible surrogate marker, and we track that marker. So the kits that we sell contain the spiking component, which is a BSL-1 virus-like particle, and the components, the additional components, are all of those necessary to perform your quantification assay. And so when you purchase this kit, the way you use it is identical to how a CRO works. You spike in, I'm trying to get the animation on the slide here. Let's see if I can figure, there we go. So you spike your BSL-1 particles into your load, just like a CRO would spike the live virus into your load. And then you pass that spiked load through your separation technique, whether that's a column, a membrane, a monolith, a virus filter. And then you can take fractions along the way, your wash, your flow through, your elution, your strip, and then you can analyze whatever you want, any of those fractions, especially the elution. That would be the basis for calculating your LRV. But if you take all the other samples as well, you can really get a mass balance where this particle is going during that separation technique and really learn about the mechanisms of viral clearance during that separation step. So yes, you take all of your collections, including your elution, and then you compare the quantity back to what was in the load, and then you can deduce your log reduction factor, which is the important data at the end of the day. And we'll talk about the analytical approaches to do that, but that's the general concept. Just very similar to a CRO. The only difference is we've replaced that live virus with the BSL-1 compatible surrogate. So now that's all the background. The next slide here is the first slide where we're going to talk specifically about retrovirus-like particles. And, when MockV and Cygnus were tackling this issue of what are we going to deliver to the industry, we had a choice. Do we make an XMULV kit, or do we make a RVLP kit, which is, when I say RVLP, that's an endogenous particle to the CHO cell line, and I'll explain that in a minute. But there's 2 different paths that we could choose. And we went with the CHO endogenous RVLP, and I'll explain to you why in the slide. But it's helpful to see a historical context here. So before XMULV became the model retrovirus used by the CROs, I'm going to take you back a decade prior to this, or 2 decades, to the 70s and 80s. And during these 2 decades, scientists were discovering budding type C particles. They look like retroviruses budding out of CHO cell lines and NF0 cell lines as well. And this was a major concern because these cell lines were just starting to be used as the workhorses for recombinant therapeutics. These were the early days of the biotech industry. CHO, NF0 were, these miracle cell lines that were just starting to be used. And so this was a startling discovery, seeing these, what looked like viral particles budding out of these cell lines. There were studies that came after that discovery, though, showing that these particles were indeed non-infectious, but still highly concerning. Here's one of the studies that you can see, 1973, mammalian cells in culture frequently release type C viruses. I'm trying to get to the next, excuse me, I'm trying to get to the, there we go. Okay, so in the 1990s, Genentech, who, they were pioneering the whole biotech industry back then, they were hell-bent on learning more about what these particles were. So they did a lot of work characterizing what they called CHO endogenous retrovirus-like particles, RVLP. And they went so far as sequencing the genome that these particles were packing. The Q5A guidelines came out in the mid-90s, and they were there to establish some guidance for viral safety. And this, this was hot off the heels of the concerns about these retroparticles. So it's no surprise that they recommend that the industry demonstrate retroviral clearance before an IND and a BLA submission. That was a direct, not correlation, but a direct result of the concern that came from these earlier findings in the Genentech studies as well. XMULV came onto the scene during this decade. It became the de facto retroviral model for demonstrating viral clearance. It's a retrovirus. It's relatively easy to propagate, and there's a plaque assay that can be readily used to quantify them. And it still serves as a very good model to track retroviral clearance. There have been no noted XMULV contaminations, I will say, that I'm aware of in the history of our industry. So here are the Q5A guidelines. Here's a Genentech paper talking about the endogenous particles found in CHO production. So Genentech never lost sight of the endogenous RVLP. They developed a quantification method in 2000 to analyze the amount of RVLP that were in their CHO supernatants. That enabled their scientists in the 2000s and 2010s to track their endogenous retroviruses through the downstream process. So these were studies that were done without the addition of XMULV. They were looking just at the retroviral particles over protein A, and then later on over polishing steps downstream from the protein A column. And they were using the method here developed to quantify the removal of those particles. And here's a couple of the papers. One interesting thing to note in these papers is that you can see the FDA was involved in these papers and others that came out during those decades. Scott Lutz and Kurt Borson mentioned here. So, let's see, trying to get, having a little trouble with the next slide. There we go. Okay, so in late 2022, not too long ago, maybe 6-7 months ago, the Q5A regulatory guidelines were revised. A draft was released, and this was the first time these draft guidances were updated since 1999. They discussed a lot of the modern approaches to purification and process development, including continuous processing, bracketed approaches to viral clearance, let's see, gene therapy, the risk mitigation strategies for gene therapy products. So a lot of things that have become relevant in the past 20 years. And they've also discussed in this draft document the possibility of using RVLP for viral clearance studies, and that implies validation. So a month after the Q5A revision the draft was released, Cygnus introduced into the market the MockV RVLP kit. And it looks like we timed that perfectly, but The truth is Cygnus and MockV before it were working on that kit since 2018. So it's kind of just dumb luck that we aligned perfectly with the release of that draft guidance. So here is the Q5A, and I just pulled out one quote that says, for CHO cell-derived products, CHO-derived endogenous virus particles can also be used for viral clearance experiments. So with that as the background for the kit, what we do here at Cygnus is we propagate CHO cells for the explicit reason of producing RVLP. We coax our cells into expressing as much RVLP as they can naturally, and then we purify the RVLP from our CHO supernatant through a 2-column process and concentrate that down into a stock solution. So the material is, it's natural to CHO. I sometimes get questions on how do we know that it's BSL-1 compatible, that the material is non-infectious, and the answer to that is we are simply growing CHO cells. We're not expressing any recombinant protein in these CHO cells. It's basically just a CHO line grown at optimal conditions for boosting RVLP production. It's no more dangerous than receiving CHO supernatant from your upstream group and then purifying it through your downstream process. That's what we're working with here. It's just a concentrated stock of these retrovirus-like particles. So we did some characterization assessments. We compared the RVLP that we're producing to infectious live XMULV. We did dynamic light scattering to track the two and also some zeta potential measurements. We also looked at the stock solution of our CHO RVLP under TEM microscope. One thing to note though in these comparisons, RVLP and XMULV are similar, but RVLP is not a virus-like particle of XMULV. XMULV is a good model to demonstrate RVLP clearance, but they are not the same virus. They are their own entities. They're in the same retroviral family. And indeed, RVLP isn't even a virus, it's a provirus. It lacks the ability to replicate or be infectious. So just to note that when looking at the slight differences you see in the characteristics. So the way that we quantify these particles is pretty straightforward. They do package a proviral genome. And that's what Genentech utilized in 2000 to establish a method. They simply extracted the RNA from these particles. They used a QIAGEN kit to do that. And then they developed a qPCR assay to quantify the amount of RNA extracted. We modified the extraction procedure. We now have a 96-well plate method, which allows for much more high throughput work. You can run 23 samples plus your standards on one plate, which is much better than using the columns from the QIAGEN kit. So, and also we've been able to lower the limit of quantification as well through this extraction technique. But for the qPCR elements of the assay, we're utilizing the same exact primer and probe set that was published on 22 years ago that Genentech has been using and publishing on with the FDA. So that's the approach that we took there. And here you see the behavior of the assay over about an 8-log window. Just note that the x-axis is in copies per microliter. To normalize that to RVLP per mil, you have to multiply by 1,000 to go from microliters to mil, and then divide by 2. It's thought that there are 2 copies of RNA per particle, so that's why we divide by 2. When you perform that calculation, this shifts the graph to the right a little bit, and it results in the limit of quantification being around 10 to the 3 particles per mil. And we'll go into a little more detail there here with the qualification of the assay. Here you see the intra-assay precision that we're able to achieve through the assay. We made a dilution series of the particle, and for each one we tested in quadruplicate, so 4 tests for each dilution point. We have the average, the standard deviation, and the coefficient of variation. So you see overall very tight data set, and then we repeated that a second day and saw an even nicer data set. But everything, below 5%. We had one data point that was a little noisy. Perhaps there was an outlier that could be statistically removed. But overall, this is a very nice, clean data set for intra-assay precision. For comparing those 2 different days, we can get our inter-assay precision. And again, you can see very nice, clean CVs over 2 different days. When you start to get to the lower concentration points, 4, 3, and 2, you start to see an uptick in the noise of the data set overall. In terms of the accuracy, as I mentioned, when we did this qualification, we prepared theoretically a dilution series at 9 RVLP per mil, 8 log 10 RVLP per mil, and so forth. The actual values that we achieved were compared back to the theoretical to get our percent to the target, which delivers our accuracy. And you see we're right on the target very, very well. When we get to the lower dilution points, though, we see another uptick in that to the extent that we start to feel a little uncomfortable about these lower points. So for those reasons, we set the lower limit of quantification to 3 log 10 RVLP per mil, or in other words, 10 to the 3 particles per mil. So this is the last slide I'm sorry, we'll see some data. But before seeing the data, I just wanted to show the kits, the components that come within it. We sell our M230 kits, which contains everything you need to do your spiking study, the particle, the quantification components, the PCR components, extraction components, your standard RNA, everything, the whole kit and caboodle right in that product. But we also sell the stock solution, which is the second, I keep forgetting you can't see my mouse, so I apologize. But the second component you see is the RVLP stock solution. That's the particle. That's sold independently. And also we sell a kit that encompasses just the extraction components, and then another kit that encompasses just the RT-qPCR components. So now let's go into some data on use of this RVLP kit. We worked with 2 biopharmaceutical companies before we commercialized the kits, and the studies that these 2 companies did spanned a very diverse group of downstream processing steps. So let's start at the beginning, protein A. We have RVLP and XMULV data to compare against. The XMULV data was accumulated at a CRO using the same lot of material, using the same parameter sets on the same separation technique. So everything being equal, these are the 2 comparable data sets. And you can see for the different phases of the protein A step, samples were collected and LRVs were compared between the 2 particle types. And you see this excellent correlation between the 2. For the protein A, the protein A elution, you see we're within 0.5 logs of predicting XMULV. And then the second group that performed this with protein A also within 0.5 logs. For virus filtration applications, you'd expect full clearance of both of these particles since these particles are large. And that is what we achieved. XMULV greater or equal to 5.3 logs of clearance. And with RVLP, greater or equal to 4.4. The reason why there's an inequality sign ahead of both of these values is that the amount of particle detected was undetectable. The value you see here is a reflection of the lower limit of quantification for the assay. It's the mathematical limit that we could claim clearance for these steps. So again, no particles were detected in these filtrates, and we were able to show the full extent of the ability for these assays to come up with these log reduction values. For cation exchange, we had XMULV value that was below the limit of detection. So there was particle, and we were able to track that as well with the RVLP to, within 0.5 logs of accuracy to that XMULV. And one other thing to note, you may have picked up on this, but the RVLP values for both the protein A step and the cation exchange step here were slightly below, in each case, the XMULV data point in each case. And, that's probably seen as a good thing. It's a more worst case value. It's a lower LRV value. So you wouldn't be over predicting the possibility or the effectiveness of that step by using the RVLP kit if you were going to compare that against XMELV. That's just an important note to put through there. With anion exchange, we actually didn't have XMELV data, but there's still a good story to tell nonetheless. When operated at center point conditions, expecting robust clearance at flow-through mode, you see we were able to achieve full RVLP clearance to the extent that the assay could deliver, greater or equal to 4.35. But when we repeated that study with a higher conductivity load and higher conductivity equilibration buffer, we saw a dramatic decrease in the LRV, which is what we expected. The salt present in that load material masked the charge, which is the basis for that separation, and the particle was no longer able to bind to the resin and came right through with the product during that flow-through collection. And that's what we intended to see there, and it panned out. That's what we saw. The group that ran this also ran their center point conditions for bind and elute method of their anion exchange, expecting full clearance, and we achieved that as well. So last slide for this. So mixed mode, we have RVLP versus XMELV. Full clearance was achieved with XMELV, and we again achieved full clearance with the XMELV as well. And then one kind of a cool thing that one of these groups did, they had their TEM results from their CHO supernatant, which most companies do prior to a viral clearance study. They want to gauge the amount of endogenous retrovirus-like particle in their CHO supernatant via TEM analysis, and that goes into the calculus that's done to calculate how much viral clearance you actually need to accumulate over your downstream process. So that's part of the math that's done, which is why companies submit samples like that. So they had the TEM value, and they used our analytical kit to also measure the amount of RVLP in that same CHO supernatant sample. And you can see that the two results overlaid very, very well. So just another data point there. Last slide on RVLP. We, from time to time, get requests from companies to purify their very own RVLP from their own process. So let me explain that. So I think I mentioned at the beginning of the RVLP section of this talk that we grow CHO cells. We have an ATCC CHO cell line that we use to produce our RVLP particle, and that's great. It's a universal stock of RVLP that we generate from an ATCC line. But some companies say, you know, we're using CHO line X or CHO line Z, and we've made modifications along the way. Can you enrich our very own RVLP from our CHO supernatant? And we can. We've done this for several companies. So they'll provide us with the CHO supernatant or even protein A flow-through. And we will put that into our process, and we will turn that around and produce their own stock solution of RVLP. During that process, we're removing most of those impurities, the host cell protein, the DNA, the therapeutic that they have in their CHO supernatants, the therapeutic aggregate, charge variants, whatever. So we give them back a highly purified, highly concentrated RVLP stock for their very own spiking uses. So now I'm more than happy to answer questions during the Q&A section of that on RVLP. But with about 10 minutes now left, let's turn our attention to MVM. So MVM, very different what we do here. For this kit, we are actually making a virus-like particle of the live MVM virus. Live MVM has one dominant capsid protein called VP2. That's what makes the structure of the particle. So what we do is we recombinantly express that VP2 protein in a system, in a baculovirus SF9 system, an insect cell line system. So the insect cells are cranking out this VP2 capsid protein. There's none of the genome of the MVM virus except for just the structural protein, which is used to build the protein. So nothing is packaged. There's no genome packaged here. We're making the protein self-assembles into a particle, and we purify that particle to form this highly concentrated, highly purified stock solution that you see this image of. And that's what we're doing here. So we're trying to do our best to mimic MVM in this case. Here you see an image of MVM that we make side by side with the live MVM produced. This MVM was produced by a CRO called Texcel, and you can see morphologically very similar. We collaborated with the FDA and asked them to demonstrate for us the size, surface charge, and surface hydrophobicity characteristics of these two particles, along with the bacteriophage that Scott Lute of the FDA was publishing on. He was using that as a guide to demonstrate the viral retentive filters as a marker, a standard, and other studies that he was publishing on use that as a marker for clearance through these types of virus filters. Anything that's size-based, he was doing with the bacteriophage. So anyway, we submitted the 3 samples to the FDA, and they used a battery of different tests to look at the characteristic similarities or differences between the 3 particle types. The methods and the full data is all published in the paper that I showed on the previous slide, but this is a very brief summary of that data. Live MVM and MVM, and our live MVM is in this middle left column. Our non-infectious MVP is in the middle right column, and then you have this PP7 bacteriophage on the very right. And you can see in terms of size, the first 2 rows, all 3 particles are very similar. Where the MVP, the non-infectious MVP, really does a better job at mimicking MVM is in terms of the surface charge and the surface hydrophobicity. You can see that data right there on the screen. The analytical assay for the MVM particle is very different because these particles don't package nucleic acid. We cannot do an extraction like we do with the RVLP kit, but we did find a way of harnessing the sensitivity of PCR. What we have to do is externally recruit a nucleic acid sequence to a sandwich that we build. For the sake of time, I think I won't go into detail here, but basically it's a 96 well plate assay. It works very similar to an ELISA up until a point. And up until that point, you add this, you can't see my mouse, but you see this antibody where there's like a tail attached. That tail is an oligonucleotide. So you break the sandwich in the immunoplate, transfer your sample to a qPCR plate, and then you can run a PCR assay off of that oligonucleotide tail. And you see on the right-hand side, this concentration dependent graph where you see the higher the concentration on the right, the lower the number of CTs, the lower number of cycles to achieve a fluorescent event. And this assay can give you about a 4 to 5 log sensitivity range, allows you to do LRVs, to calculate our LRVs up to about 4 to 5 logs. So here's a picture of the kit. It contains everything you need to run your study, and we provide every single one of these components individually as well. So some customers purchase additional spiking stock to boost the amount of particle that they put into their load. So with the few minutes I have left, let's look at a couple data sets. Maybe I'll just get through one of these, and then if a question pops up, I can refer back. But the NIH actually came to us. They were doing some process development work utilizing their high throughput TCAN instruments, and they wanted to look at viral clearance over a diverse range of different resins, all provided to them in robocolumn format from the different resin vendors. So they can't take their TCAN into a CRO to do a viral clearance study, and they can't get live virus into their lab because that's a big no-no. So there really is not an option to utilize the TCAN in a viral clearance type of setting unless you use an approach like the MockV approach. So this was kind of like tailor-made for this sort of work. And I'll show you the next slide. It's very busy, but let me explain. So on the very left-hand column, you see all the different resins that the NIH was interested in exploring. These are all anion exchange resins. For each resin, they equilibrated their columns to 3 different pH points. And then they equilibrated their load materials containing their vaccine candidate to these 3 different pH points. They introduced our MVM particle at a known concentration into the loads. and then they applied these loads to these robocolumns, spun down the flow through, collected a sample. They then exposed these resins to 50 millimolar salt buffer, spun it down, collected another set of samples, and so forth and so on, collecting samples in between each incrementally higher molarity salt exposure. And you'll see some of these resins had the ability to retain the particle into these salty conditions. These values you see are log reduction factors. So the 4.8s you see are indicating that these particles were still being held, and in these flow-through collections, none to little particle was being found there, leading to these high LRV values. But the lower values are indicative of particle breaking off of the resin and being present in these collections when exposed to these higher and higher salt conditions. So you see some of these resins behave extremely well. Poros 50 pi and Toyo-Pearl NH2 750F in particular. That's what the NIH was looking for here. This was the data that they were hoping to achieve. They wanted some guidance on what resins were working best for their process step. And this is what they were able to get. So that whole study was published in a biotechnology journal. They looked at cation exchange as well. And it was just a really nice data set. So I'm not going to go into detail on these slides because I want to save time for Q&A, but we've done work in the gene therapy space. If anyone's interested during the QA, can go back to these slides. We also have the data I was going to show is also published. So if anyone wants to reach out to me after the talk, I'll be more than happy to just send the paper. And the last slide I do want to review is the analysis service that we offer. So often the case is that the purification teams are very excited about these kits, and they see, the use for these in the downstream process development and characterization space. But the analytical groups aren't so keen on bringing on board another assay. So it sometimes falls on the purification group to run the assay. And that's great. We'll help you 100%. We'll do video conferencing with you, Teams, Zoom, whatever. We can even come on to site and help you establish the assay, work with you on the bench to help you establish these assays. But if you want to just prove the technology before going through all that rigmarole, you can purchase just the particles. We'll ship the particles to you. You'll perform your study and save your samples. You can ship the samples back to us. We'll perform the analysis. We run these assays every day, and we'll generate a report. We'll show you that report. If the data is to your liking and it's something that you think is of value to your team, then maybe it's something that you want to internalize. Maybe it's we could come on site, train you up on the assay, or you're like, this is great, service model works great, let's do that again. We work it both ways. So just wanted to make that known for both of the kits that we sell. We run that service model as well. So let me turn it back to Jeannie now for Q&A. Thank you for sticking around 50 minutes in, and looking forward to the Q&A session.
David, thank you. You can tell this is a field that you are a proven expert in. I found your presentation to be very supplemented. I love the data. It really explains what all is available to help support these studies. So thank you.
I appreciate that.
I'm also seeing a lot of great questions coming in from the audience, so I'm excited to get started. However, before we start, for anyone who may have logged in a little late, I just want to remind our audience how they can submit a question to David. I'd also like to let you know that while we are running out of time quickly, David will review any questions that are not responded to today offline, and we'll be able to respond offline to you as well. You can submit your questions by typing them in the Q&A box, and that can be found directly below your video player. So that said, I'm going to pull up some of our questions, David.
Sure.
And okay, here's a good one, David. Can the RVLP kit, going back to RVLP, be used for low pH inactivation studies?
The answer right now at this moment is unfortunately no. The mechanism by which low pH inactivation degrades a retrovirus is such that the envelope dissipates, and that's true with our RVLP, but the underlying capsid, which protects the RNA, is still there. And so with our assay, you would still get a positive result, leading you to believe that you have intact RVLP, when in fact the envelope has been degraded. And that's why with the plaque-based assay, there's a failure to show that type of particle. So there is a disconnect right now with XMELV versus RVLP for low pH inactivation. That being said, there's so much interest in this product for low pH inactivation. We're working with a large biopharmaceutical company in working out an assay which can mechanistically skirt around what's going on there and still provide useful data. So perhaps in the future, in the near future and midterm future, we'll be able to deliver that. But at this time, no.
David, maybe real quick, in a short answer, briefly, you can describe what the shelf lives are for our audience for both MVM and the RVLP particles.
Absolutely. So the MVM particle is extremely hardy, as is the virus that it's serving to mimic, MVM. Parvoviruses overall are very tough viruses. So we have data that supports over 2 years of stability when held at 4 degrees for the MVM particle. RVLP is a different story. Those particles are known, retroviruses overall are known to be less stable over time, and we see that as well with our RVLP. We've proven out a 6-month shelf life when stored at 4 degrees for the RVLP stock solution.
Thank you, David.
Sure.
Have these mock viruses tested in any multi-column chromatography yet?
Multi-column chromatography. No, not to my knowledge. They may have, but I have not seen a data set yet
Sure.
to that extent.
Sure.
Yeah.
Okay, let's talk about some of the controls you have in place. I know you said you provide so many services in-house as well as out, and then we think about your solutions. So what kind of controls do you have in place to make sure that your RVLP kit is not contaminated with any live? I know you started to talk about the screening for the live viruses from CHO and the cell production process.
Yeah, no, that's a great question. So, I mean, Cygnus has a robust quality system. We abide by that quality system to the fullest extent. We cannot 0.22 filter the RVLP stock because the particle itself is borderline pushing 0.2 microns. So we would be losing our product. So what's standard and typical for, let's say, lentivirus, we borrow a lot of theoretical knowledge on how to produce from the lenti space. It's very similar. We 0.45 filter our particles. We use, I don't want to call it a preservative, but an antibacterial chemical in the final formulation to prohibit the growth of bacteria, mold, things of that nature, yeast. How do we make sure that there's no viral contamination? That is a good question. We do submit at random stock to look at under the microscope, and that's true of MVM as well. Every batch we make of MVM, we submit for TEM analysis, and TEM analysis is very good at spotting any contaminations of the viral type. So there, in the past, back in like 2017, 2018, we did have a couple MVM batches we produced that had residual baculovirus present. And we implemented a second chromatography step in our process to get rid of the baculovirus. And that's been in effect ever since. And since we've implemented that, we've never seen baculovirus or residual baculoviral debris in any of our MVM preps. So that's one thing we're doing with the MVM and the RVLP kits there.
Very promising. And, I do have an urgent question that I want to get to, but beforehand, just because you started to talk a little bit about resin reactions, are you aware of any resins where MockV particles they don't work well? When compared with other question. Yeah, any resins that maybe there's no resin that I can say it doesn't work with. I've seen very good data sets for all resin types that I'm aware of. So I'd say no, but I mean, in full transparency, 10% of
customers that we sell kits to, maybe 5 to 10% say, hey, I'm getting results that are not correlating well with my MVM data. I haven't seen any of those emails come back from RVLP yet, but MVM has been on the market longer. So from time to time, we get some results where the data supposedly isn't correlating very well. It's not particular to a certain type of resin, though. In those cases, it's hard to say. It could be maybe there's something incompatible with the material, the in-process material that we're working with, or maybe there's something nuanced with the assay, the plaque assay versus the immuno-qPCR assay. They're two very different types of assays, and we're trying to compare data across them. So it's a hard thing to try to do, and it works the vast majority of the time. But in full disclosure, it doesn't work 100% of the time. So to answer your question, it's not particular to any type of resin per se, but it should be known that there is that possibility that for your material, there may be a disconnect. Hopefully not, but it's possible.
Great, thank you. Has that MockV approach to viral clearance, has it been implemented in a BLA filing? The
use of RVLP may have. I was presenting to a group 3 weeks ago, and I was presenting that slide where I take you through the history of RVLP. And, clearly you could see that pattern there. Genentech has been pushing the use of RVLP for 30 years almost. And they may have filed a BLA filing with RVLP as the basis for retroviral clearance instead of XMELV. I didn't think that was the case, but the group that I was presenting to seemed to think that it was. So I guess there's a little controversy on whether or not a company like that has actually gone through and done that. So, but if they haven't, you can see the trend that our industry is going into. The Q5A guidelines are making note of the possibility to use that approach. It's being discussed at every viral clearance safety forum and presentation and conference that I've been attending for over a year or two now. So with the RVLP kit that we're offering, we're hoping that just kind of pushes the envelope. I think the FDA is open to that possibility if the right controls are in place, the right discussions have been had, everything has been thought through to the fullest extent. I think that will set the stage for the proper use of RVLP to be enabling for IND or BLA studies now, possibly, and certainly in the future.
Thank you, David. Yeah. I have time for one last question, and just because we started to talk a little bit about regulations, someone in the audience would like to know if your RVLP detection kit replaces virus quantification by anything else from a regulatory standpoint.
Yeah, I mean, that's kind of what I was just touching on. I don't know what more to add, though. I mean, it could. I think in the near to midterm, as the customers, as you all start utilizing this kit and presenting the data and talking to Scott Lute, talking to his lab at the FDA, presenting the case, they're very much into sound data, and they're aligning with easing viral clearance if the data is there to support it. They've always been supportive of new ideas if the data is there to support it. I'll be presenting at the PDA forum in Europe next week. I think it's well established that the EU is a little more conservative when it comes to viral clearance, but I'm going to be pushing this concept there, and we'll start chipping away at the mind state. I do believe that XMELV is a model. The model is for endogenous retrovirus particles. That's what we're providing here in this kit. So it's flipping the mentality, which has been established over 20 years. But I mean, think about that. It's a model virus. That's what XMELV is. So I think the case is solid.
It is a good case, and it's something that we're hearing so much about in research efforts in healthcare anyway, right?
Right.
So again, David, thank you for such a great presentation today, and thank you so much for your time.
Thank you all. Thank you, Jeannie.
Yeah. As we close out and wrap up this program, I'd also like to thank our sponsor, Cygnus, for making today's educational webcast possible. And I'd like to thank our audience for attending and for reaching out to David with some really great questions. Again, for any questions that were not responded to today, which I saw quite a few still on the list. David will review and respond offline. As we close out, I'd like to ask everyone in the audience if they would kindly participate in a brief survey. The survey will appear on your screen just after this presentation has concluded. You'll also receive an email alerting you when this webcast is available for replay, and we ask you to forward that announcement to any colleagues who may be interested and may have missed today's live event. So thank you to everyone for joining, and we'll see you next time. Bye-bye.
Thank you.